ever wondered how your brain's incredibly complex activity, all those millions of neurons firing, how that actually translates into measurable electrical signals?
Yeah, things like EEG that we see on the scalp.
Exactly. EEG, or even the local field potential, the LFP, which is sort of listening closer right inside the brain.
Right. Understanding that jump from the tiny scale of neurons to those big macroscopic signals, well, it's been a real puzzle in neuroscience.
It really has. So today we're doing a deep dive into a really interesting research chapter. It's called On the Electrodynamics of Neural Networks.
This is intriguing.
It is. And our mission really is to unpack this new model they propose, because it seems to finally bridge that gap, connecting individual neuron activity to these large-scale brain signals.
Kind of like a missing link.
Kind of. Think of this as your shortcut to getting up to speed on one of the brain's big mysteries.
Great, let's get into it.
Okay, so let's set the stage. This challenge, it goes way back, doesn't it, to Hans Berger, who first discovered EEG.
That's right, 1924, a long time ago.
And ever since then, the big hurdle has been linking that sort of hidden activity of single neurons to the bigger picture, the LFP, the EEG.
Right.
How do tiny flickers become a wave?
And it turns out the main source, the generators for these signals, are specific cells, the cortical pyramidal cells.
Okay, why them specifically?
It's their shape, their structure. They're like little trees with synapses, input points up high on the apical branches and down low on the basal ones.
Right.
So when they get input simultaneously in those different spots, it creates a tiny electrical separation, like a mini battery, a microscopic dipole.
Ah, okay.
And these tiny fields from the dendrites, the DFPs, they all just add up. They superimpose.
It's like ripples in a pond joining together.
Exactly, forming that larger wave, the LFP. And that LFP, in turn, generates the EEG we can measure way out on the scalp.
Okay, that makes sense. But you mentioned earlier models struggled with this.
They did. Big time. Connecting the discrete neurons to the continuous fields was tricky.
How so?
Well, often they just ignored the extracellular space, the fluid around the neurons.
Oh, right.
Or, if they did include it, they treated it too simply, just as like a resistor, like current flowing through a simple wire. They missed something crucial.
Which was?
Diffusion. This paper really highlights that diffusion currents in that extracellular space are essential.
Diffusion currents. You mean ions just moving around based on concentration, not just voltage?
Precisely. Ions moving down their concentration gradients. And it turns out this contributes significantly to the frequency patterns, the power spectra we see in these brain signals.
Wow. Okay, so it's not just the direct electrical firing, it's also this background ionic flow.
You got it. It's a key part of the picture that was largely missed.
So how does this new model incorporate that?
Well, the authors use what they call a microscopic approach. They have this 3-compartment model for the pyramidal cells.
Okay.
And the really clever part is it explicitly includes both the resistivity and the diffusion in the extracellular space.
Yeah.
They use the Nernst-Planck equation to model how ions actually move.
So much more biophysically realistic.
Exactly. Much more grounded in the actual physics and chemistry happening there.
And how does this detailed single-cell model help with the bigger picture, the whole network?
This is the elegant part.
Yeah.
This detailed model can be simplified mathematically into something called a leaky integrate and fire, or LIF model, a standard way to describe basic neuron behavior. And then, using a technique called the continuum limit, they scale it up. It evolves into an Amari-type neural field equation, which describes large populations of neurons.
Okay.
And crucially, it's coupled with Maxwell's equations, the fundamental laws of electromagnetism.
Wow. So it links the biology, the neuron behavior, all the way up to the physics of the electric fields.
Precisely. It's presented as the first model that's truly biophysically plausible and consistently explains the widespread dipole assumption.
That assumption being that these brain signals mainly come from those tiny dipoles we talked about.
Yes, exactly. It's a cornerstone idea for interpreting EEG and MEG, but this model provides a solid grounding for it, starting from the single-cell level.
Okay, so the theory sounds impressive, but does it actually make better predictions? Did they test it?
They did. They compared their models' LFP predictions, they call it 3L33, against a widely used older model, the Midjourney et al. Model, or LF3. The difference was pretty stark. The older model, LF301s, systematically overestimated the LFP amplitude.
How much?
By almost 1 order of magnitude. It's nearly 10 times too high.
Wow.
And it also got the frequencies wrong, predicting signals that were too fast compared to reality.
So this new model is much closer to actual measurements.
Yes, their model produces LFP estimates that are much smoother and, importantly, comparable with experimental LFP measurements down in the millivolt range. It matches reality much better.
And that accuracy must be pretty important.
Hugely important. For clinical applications like understanding epilepsy or improving brain-computer interfaces, you need models that accurately reflect what's happening.
Right. Better models mean better understanding, potentially better treatment.
Exactly. It helps bridge that critical gap between the underlying neural activity and the signals we can actually measure and work with.
So wrapping up, we've really seen a significant step forward here. Understanding how neuron activity generates these brain signals, really nailing down the role of neuron shape, and kind of surprisingly, the importance of these diffusion currents.
Yeah, that diffusion insight is really key. It adds a whole new layer to how we think about the extracellular space.
It's a huge advance.
It is, but it also opens up new questions, doesn't it? It reminds us there's still more to figure out.
Like what?
Well, what happens when we start accounting for things like polarity reversals? You know, how the signal flips as you move an electrode through different brain layers?
Right. Okay.
Or what about ephaptic interactions? That's where neurons can influence each other electrically just by being close without needing a synapse. That adds another layer of complexity.
True. Direct electrical crosstalk.
Exactly. So while this model is a fantastic leap, the brain's electrical symphony, well, it definitely still holds some secrets. We're getting better at listening, but there's more to uncover.